the ongoing lecture series on plant tissue culture, we were discussing about various steps that are involved in a plant tissue culture experiment. In that, we have already discussed about the cleaning and sterilization of glass and plasticware that are to be used in the experiment. Then we discuss about various components of the plant culture media. So depending upon your requirement, you can select various components to be incorporated into the culture media. Then we discuss about the preparation and sterilization of the culture media which you are going to use for your experiment. Then we started with the selection of the explant material, that is, what is an explant, what are the various factors that affect the selection of an explant. And friends, today I am going to start with another step, that is sterilization of the explant. As we discussed in yesterday's lecture, that plant tissue culture is a technique which requires utmost conditions of sterility. But there are chances that contamination may get incorporated into your study through the explant material if it is not properly sterilized. Now, sterilization of the explant largely depends upon what type of sterilizing agent you are using and the duration of the sterilization. Now, the type of sterilizing agent as well as the duration of sterilization will also be governed by the type of the part which are using as an explant. By and large, in majority of experiments, it has been seen that chlorine bleach, which is sodium hypochlorite, it is widely used as a sterilizing agent in a concentration range of 3 to 10% in water. Now, this percentage is for the commercial bleach solution. If you are using pure sodium hypochlorite, then 1 to 2% in water is sufficient for sterilization. Now, apart from chlorine bleach, various other agents are also used for sterilization, and these are 70% ethanol, calcium hypochloride in a concentration range of 0.1 to 1% along with 0.5% hydrochloric acid, hydrogen peroxide in a concentration range of 3 to 10%, mercuric chloride 0.2 to 0.5%. Now, this agent is highly corrosive and is only used for highly contaminated samples because majority of the explant material are very sensitive to mercuric chloride, and if you have to be very critical regarding the duration of exposure of the explant towards the mercuric chloride. And last but not least is the silver nitrate in 1% concentration. Now, as I told you that sterilization duration as well as use of sterilizing agent will be largely dependent upon the type of the explant you are using. Now, if you are using an explant material that has been obtained from the subculturing of a callus or a suspension, that is, that explant has been grown under aseptic conditions in vitro conditions, obviously that explant would be highly sterile and you can use it. Say for example, if we are using seeds as explant, so first we have to wash the seeds with detergent and rinse them with fresh water. Then we will treat those seeds with 70% alcohol for around 2 minutes, followed by the treatment with sodium hypochlorite solution 20 to 40% of the commercial bleach product and 1 to 2% of the pure sodium hypochlorite solution for say 15 to 20 minutes. Then you can also add 2 to 3 drops of tween 80 to the sodium hypochlorite solution because this tween 80 will act as a surface acting agent or surfactant which will improve the wetting of the seeds or improve the contact of the seeds with the bleach solution, thereby enhancing the sterilization ability of that particular solution. Now, once the treatment with sodium hypochlorite solution is over, under the laminar flow we will decant this chlorine bleach solution and wash the seeds with sterile or demineralized water for 5 to 6 times. Chlorine gas can also be used to sterile the seeds that have to be used as an explant, but it has been found in numerous studies that it reduces the viability of the seeds and therefore reduces the growth ability of the seeds onto the artificial culture media. Many times you will see that certain plants provide you hard coated seeds, like clover is there, alfalfa is there. For such seeds you have to subject those seeds towards scarification process, that is removal of the hard testa, that is seed coat, by just rubbing those seeds on a rough surface or by a slight grinding of the seeds and then sterilizing them. Many species have seeds which are under dormancy, and therefore they are required to be subjected to a process which we call as vernalization, that is breaking of dormancy. Now, vernalization is a process, is a term that has been derived from a Latin word vernus, and the vernus means spring, and we know that in spring season there is maximum cell growth ability, maximum cell division capability, and spring normally follows after a winter season. Therefore, to break the dormancy of such seeds, they are stored in a room at a temperature of 4 degrees Celsius for 25 to 35 days. Now, by exposing those seeds towards this longer cold durations, it helps in decreasing, it helps in breaking the dormancy of the seeds, and whenever we are sterilizing those seeds and placing them onto the culture media, they start dividing, the cells start dividing in the seeds. Now, if you are using the aerial parts of a plant as an explant, aerial parts, we know that they include leaves, shoot bud or stem parts and so on and so forth. Now, to sterile these aerial parts, we will submerge these parts for 1 to 3 minutes under 70% ethanol, and then the tissues are submerged in 20% commercial bleach or 1% pure sodium hypochlorite for 5 minutes, and then we will rinse these parts for 3 to 5 times in sterile or demineralized water. This is how you sterile the aerial parts if you are using them as an explant. Now, we move on to the underground parts like tubers, roots and bulbs. Now, when we talk about such underground parts, we can assume that these parts will be highly contaminated with mud or soil. So, therefore, in order to perform the sterilization of these underground parts, first we have to wash them with detergent in order to remove any kind of mud or soil that is adhered to these parts, and then we will rinse them through using the running tap water under high pressure, so that if any soil still remains after washing with detergent, it gets removed by the high pressure water. Then these parts are submerged in 70% ethanol for 1 to 3 minutes, and then the tissues are exposed to sodium hypochlorite solution or commercial bleach at the required concentration for 20 minutes. Now, we can always repeat the treatment with chlorine bleach if required, if the underground parts we feel that they are not completely sterilized, we can repeat the process. Once the treatment with chlorine bleach is over, we can rinse those tissues or we can rinse those parts 4 to 5 times with sterile or demineralized water. If you observe that any of the tissues, whether they are seeds, whether they are aerial parts or whether they are underground parts, if they get a brownish color appearance or whitish appearance on their surface, this will indicate that they are over sterilized and the cells are dead. Now, if we see that a particular area of the explant material is over sterilized, then we can always cut that portion and we can use the remaining explant. If an explant, specifically in case of underground parts like roots and rhizomes, they are highly contaminated and we are not able to sterilize them by the normal sterilization process which we have already discussed, what we can do is we can cut those roots and rhizomes into multinodal segments. Now, these segments are then treated with gibberellic acid solution in the required concentration, generally 50 to 100 milligram per liter of concentration of gibberellic acid solution is used. Now, by this treatment we are promoting the growth of buds on the roots and rhizomes. And these buds now can be used as explants in place of the entire pieces of roots and rhizomes. So these are some of the examples how you can change or how you can manipulate the sterilizing procedure if you are dealing with different types of explants. Now we move on to the next step in the plant tissue culture experiment, that is preparation and sterilization of the inoculation area. Now you have prepared and sterilized the culture media, you have prepared and sterilized the explant. Now the next step is to prepare and sterilize the inoculation area where you are going to place that explant onto the culture media. Now in order to prepare the inoculation area, first of all we have to turn on the laminar flow which is fitted with HEPA filters in the transfer hood to maintain a positive air pressure. Now this positive air pressure is important because whenever you will open the lid of the transfer hood, the air should flow from the transfer hood towards outside. We cannot allow the contaminated air outside the transfer hood to flow inside the transfer hood. Therefore, this positive pressure is always maintained. Then we will spray the interior of the transfer hood with 70% ethanol, which will help in causing the denaturation of the DNA, bacterial DNA, thereby killing the bacterial cells. Anything that is placed inside the transfer hood should also be sprayed with 70% alcohol. Spray on HEPA filter should always be avoided. Now when you will see in the transfer hood, there are numerous things that are placed inside the transfer hood, like there would be sterile wrapped bundles which will include all those tools like forceps are there, scalpels are there, spatulas are there, which are required to cut the explant, which are required to hold the explant and transfer onto the culture media. So everything that is placed, the working tile on which you are going to place the explant and cut the explant, which is already pre-sterilized and wrapped in an aluminium foil, so everything has to be sprayed with 70% ethanol. We can also saturate a piece of a cheesecloth in 70% ethanol and wipe the entire area of the transfer hood from inner side with that ethanol. This will also help in sterilizing the surfaces of the transfer hood. So once we are done with the treatment of the transfer hood with 70% ethanol, we will wash our hands with detergent. We will then spray, we will remove any kind of jewelry that is there in our hands, like rings are there, bangles are there. So if they are present on your hands, you have to remove them and then spray it with 70% ethanol before you enter your hands into the transfer hood. Once everything is sterilized, then we will open our working tile by removing the foil, and the air from the laminar flow should laminar hood should flow across the surface of the tile, and the flow of the air should not be blocked. Whatever open containers are there, they should be placed back as much as possible into the transfer hood. The opening of the container should always be passed through the flame, which will help in creating a warm updrift of an air current, and this warm updrift of an air current will prevent the contamination of those open vessels that are placed in the transfer hood. Now when we are, as I told you that there are certain wrapped bundles of the pre-sterilized or autoclaved tools like forceps are there, scalpels are there, spatulas are there. What we will do is we will open those wrapped bundles and we will place all those pre-sterilized tools into a jar that is filled with 80% ethanol. Whenever we have to use these tools, like if you want to remove a spatula or you want to remove a forceps from that jar, you will hold the forceps in such a manner that the tip of the forceps will angle downwards. Now this will help in removal or flowing of excess of ethanol away from your hands. the ethanol will not flow onto your hands or your fingers, it will flow downwards back into the jar. Once the excess alcohol is dripped back into the jar, we will pass that forceps through the flame. Now this flame will help in consuming the excess of alcohol which remains on the surface of the forceps. So once this is done, now our tools are ready to be used for the treatment or are ready to be used for cutting or transferring the explant onto the media. Many a times it has been observed that Bacillus macerans is a species, is a bacterial contaminant which has been found to be viable onto the forceps or scalpels or spatulas after being stored in 95% ethanol for several weeks. And it has also been found that remains of this bacterial contaminant still remain after flaming of those tools. Therefore, we should always pre-autoclave our tools at the prescribed conditions, that is 121 degrees Celsius for 20 minutes, or by heating for 6 to 8 seconds over the flame before we put them into the transfer hood. And that is why I told you that pre-sterilized wrapped bundles are placed in the transfer hood, which are nothing but the tools have already been washed and autoclaved, and they are wrapped in the sterile paper before they are transferred into the transfer hood. Now once your inoculation chamber or the transfer hood is completely prepared and sterilized, we move on to the next step, that is inoculation or transfer of the explant onto the media. We have already selected the explant in one of the previous steps. We sterilized the explant. Now in this step we will transfer it. If we are using test tubes as the culture vessel, then we will transfer the explant using the forceps onto the slants of the media. We discussed that the media should be set into the test tube in the form of slants that will help in increasing the surface area of the culture media to which the explant is exposed. If we are using flask or jar, we can simply place the explant onto the surface of the culture media. Now this transferring is obviously done under the aseptic conditions under laminar flow in a transfer hood which we have already sterilized in the previous step. Now once this transferring of or inoculation of the explant onto the media is done, the containers are then transferred into the BOD incubator or bioreactors or into the culture rooms which have set under required temperature, light and humidity conditions. So after transferring the explant media, the next step is the maintenance of those cultures and subculturing of the culture. Now to maintain those culture, sterile conditions of the required temperature, humidity and light are maintained in the culture rooms, because after transferring or inoculating the explant onto the culture media, all the vessels, if you have BOD incubators, you can transfer them into the BOD incubators with prescribed temperature, humidity and light conditions. If you don't have incubators, we have culture rooms, then those sterile culture rooms should be maintained at the required temperature, humidity and light conditions. So we transfer those culture vessels either into BOD incubators or in the culture rooms. Then once we feel that or after a specified period of time when the nutrients of the culture media are exhausted, then subculturing is done. The cells or the cultures that are grown on a culture media, if the media is being exhausted, we replace that media with the fresh media, or we transfer those cultures onto the fresh media. This is largely done, as I told you, that due to depletion of the nutrients in the culture media, loss of water in the culture media and accumulation of the toxins in the culture media, because cells are growing, the normal metabolic pathways are going there inside the cells which release various byproducts and which are toxic towards the growth of the cells. So all such toxic or growth inhibitors are removed by changing the culture media, which we call it as subculturing. Many a times the type of the contamination, if you observe it onto a culture media, that will give you an indication the problem area, or you can easily identify the source of contamination by just having a visual inspection of the contamination that is occurring in your cultural experiment. It has been seen that if you find the contamination just around the explant material onto the media, this will indicate that your explant was not properly sterilized, since the contamination is localized around the explant material. If you see the contamination has been spreaded all over the media, then that indicate the poor overall technique or improper sterilization of hood, improper sterilization of a media that has been used in the experiment. If you see that your media is fuzzy or there is a thread-like growths in the media, that will indicate a type of fungal contamination in the media. The color of the contaminated area also give you an indication about the type of contamination. Like if you have smooth pink, white or yellow colored contamination areas onto your media, that will indicate a bacterial contamination. If you see striations on your culture media, lines on your culture media or tracks on your culture media, that will indicate there is an insect in the transfer hood, and that is an insectal contamination. So this is how, by observing the type of contamination, the characters of the contamination, you can easily figure out the source of the contamination. So friends, these were the different steps that are involved in performing a plant tissue culture experiment, right? Beginning from the cleaning and sterilization of the glass or the plastic where you are using, by studying the different components of the culture media, and according to your requirement you can select the appropriate components of the media. Then how to prepare the culture media, what are the various precautions you have to take while mixing the different ingredients of the media, then how to sterilize the media. The next step was about the selection of the explant material. What are the factors like age of the explant, whether it is young or old, season of the collection of the explant, what is the genetic quality of the plant material from which explant was collected, and how to sterilize the explant. Then we studied about the preparation and sterilization of the inoculation area, wherein the transfer, how to sterilize the transfer hood where you are going to transfer or inoculate the explant onto the culture media. After that, we discussed about the inoculation of the explant and maintenance and subculturing of the culture which you are growing. So friends, in the next lecture we will discuss about the different types of the cultures which you can perform. Thank you.